Hey guys, we're solving this bio paper today. This is the last paper in the paper 1 series from May June 2022, 9700 paper 13. Let's take a look at the threshold. So A was at 31, B was at 28. So comparatively this was the easiest paper out of the rest. Out of 40 marks, C was at 24. Okay, 21 and 18. So yeah, compared to the other papers, this was apparently easier. So let's begin. Starting with question number 1. Also guys, make sure to subscribe to the channel. It will help me a lot, and like the video as well. That helps with the algorithm a lot. Good luck for your exams, by the way, on the 16th. Hopefully it goes well. Hoping to wrap this up within 1 hour. Number 1. The diagram shows a section through epithelium found in part of the respiratory system. Clearly that's a goblet cell. What's the magnification of the diagram? We know that the formula is m is equal to I by a. So what you have to do is, I don't have it printed over here. You have to measure that 80 micrometer mark with a scale. Okay, so I'm gonna go backwards. I know the answer is B. 315 to 18 to 10.6. Okay, so if you have it printed, this length should be equal to, this length over here should be equal to 2.8 centimeters. So this is the working. M is equal to image length 2.8 into 10 to the power minus 2 meters divided by 80 micro is 10 to the power minus 6 meters. So 2.8 into 10 to the power minus 2 divided by 8 into 10 to the power minus 6 gives you an answer of 3005. I messed up a little bit. It's fine. 2.8 into 10 power minus 2 divided by 80 into 10 to the power minus 6. Yeah, that gives you an answer of 350. Okay, so the magnification is 350. It should be about 2.8 centimeters. First students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. mRNA passes through the ribosome. We've seen this in paper 1.1 and 1.3. It's quite similar. It should be pretty easy. So this is basically a double membrane interspersed with pores. This is the nuclear envelope. So one should be X. Synthesis of polypeptides. This is ribosome. It's a non-membrane bound spherical structure. This is ribosome. Packaging of hydrolytic enzymes. This is the Golgi body. Golgi body. We had this in paper 1.1 as well. So the last one is actually Z. So 2 is actually C. And the second one is ribosome W. What about the other ones? This is central. It's in charge of making the mitotic spindle, if you recall. And this is basically the smooth ER. Okay? Synthesis of lipids. 3. This is also another common question. An experiment was carried out to separate the cell structures in an animal cell. The cells were broken open, the extract was filtered and put into a centrifuge tube. This tube was then spun so that the heaviest cell structure sank to the bottom first, forming pellet 1 as shown. The liquid above pellet 1 was poured into a clean centrifuge tube and spun again at a higher speed to separate the next heaviest cell structure. This cell structure sank to the bottom, forming pellet 2. This procedure was repeated twice to obtain pellet 3 and pellet 4, each containing a single cell structure. So you guys need to understand something. Okay, you guys need to understand something. Like, what is the basis of separation of these pellets? Mainly in the centrifuge machine. What happens is the heaviest one The heaviest one will sink to the bottom first. Okay, so if we think about these guys, production of ATP, this is the mitochondrion. See, this is a protein, this is the ribosome. Digestion of all organelles, this is the lysosome, basically. Production of mRNA. Basically, the heaviest one sank to the bottom first. So the heaviest one, production of mRNA. Basically, mRNA production and RNA production. RNA has two components: rRNA, ribosome RNA, and protein. So this is mainly produced by the nucleus. So this is pellet 1, okay? This is pellet 2, this is pellet 3, and this is pellet 4. You will see these in other questions, okay? So the heaviest one is the nucleus itself, C. Okay? For ATP molecules are synthesized in mitochondria. Which sugar is found in these ATP molecules? Basically, you need to know that ATP has ribose, adenine, and 3 phosphates. So we have ribose, not deoxyribose. These are wrong. These are hexose sugars. So we have pentose sugar, which is ribose specifically. Which row shows a comparison that is not correct between a typical prokaryotic and typical eukaryotic cell? The DNA is not associated with histones in prokaryotes. True. Eukaryote, it is eukaryotic plant cell. It is true. No endoplasmic reticulum present in prokaryote. True. Endoplasmic reticulum is present. In prokaryotic cell, peptidoglycan cell wall. This is true. In bacteria, we have peptidoglycan cell walls, and in plant cells, we have a cellulose cell wall. These look true. So D should be the answer. All ribosomes are approximately 18 nanometers in diameter. All ribosomes are typically 22 nanometers in diameter. This is wrong. Because in plant cells, we also have 70s ribosomes, okay? That's the tricky part. So did they mention the diameter of the ribosome in the course book? So basically 80s, and I don't think I have chapter. Oh, I do. Ribosome. They didn't mention it, right? So it's 22 and 18 apparently. I know that it's around 20 nanometers approx, the 80s ribosome. So yeah, the 70s would be slightly lower. But yeah, you guys need to understand that eukaryotic plant cells also have chloroplast, which is a prokaryote. So 18 nanometer ribosomes would also be present there, okay? So D is wrong. It is suggested that primitive prokaryotic cells may be ancestors of certain organelles in eukaryotic cells. Which organelle is most similar in composition to a typical prokaryote? Mitochondria or chloroplast, okay? 6C. The concentration of reducing sugar in a solution can be found if an observational measurement is compared to a standard. Which observational measurement could be used to estimate the concentration of reducing sugar in an unknown solution? Okay, the color of the solution after 20 minutes, that works. The time for the first color change to occur, this is also true, like how long it takes for the color change to occur. The rate of formation of solid particles. Honestly, this isn't a measurement technique. 3 is actually wrong, it should be 1 and 2 only. We mainly measure time, because to measure rate, we need to find out the change in solid particle per unit time. That would be impossible to observe, okay? Since this is an observational measurement. The diagram shows 3 hexasugars, which row correctly shows examples of carbohydrates in which these 3 hexasugars occur. Okay. So remember, in alpha glucose, alpha is like this, the OH group is here. For beta glucose, the OH group is above. So 1 is actually alpha glucose. Okay? And 2 is actually beta glucose. So in cellulose, we'd get beta glucose 2. In cellulose, we'd get beta glucose 2. Now, 3 is actually fructose. So we'd get 3 in sucrose. That's why I'm going to go with D. And 1 is alpha glucose, which is present in amylopectin. Trehalose is a sugar that gives a negative result when tested in Benedict solution. So it's basically a non-reducing sugar. Which way is correct? So it has one. It is deficient of one water molecule. Since we formed a glycostatic bond, it's between A and B. Sugar that gives the same result in the Benedict's test. Clearly sucrose, right? So 9 should logically be B. Olestra is an artificial lipid. It is made by attaching fatty acids by condensation to a sucrose molecule. A simplified diagram of Olestra is shown. R represents the position where fatty acids would be attached. Okay. Humans cannot hydrolyze Olestra, however, other mammals may be able to do so. How many molecules of water would be needed to hydrolyze one molecule of Olestra into fatty acids, fructose, and glucose? Interesting. So guys, look at the answer. Basically, there will be one hydrolysis here since the R is a fatty acid. One over here. 1, 2, 3, 4, 5, 6, 7, 8. That's 8 fatty acids. Fructose and glucose. So this glycostatic bond over here. So in total 9. 10C. Great. Moving on to 11. Which molecule contains at least one peptide bond? A is a triglyceride. C is basically disaccharide. It's between B and D. What is B? Look at this. CONH. 11B. Peptide bond. D is basically part of DNA. 12. RNA polymers and peptidyl transferase are both enzymes involved in protein synthesis. Which statements describe similarities between the two enzymes? They are both globular proteins. This is true. They are enzymes, globular proteins. They both have the same tertiary structure. Come on. This doesn't make sense. How? They are not the same enzyme. So 2 is wrong. They are both intracellular enzymes. This is true. They work inside the cell. RNA inside the nucleus and peptidyl transferase in the cytoplasm. Okay, so for 13, what is a feature of competitive enzyme inhibition? Basically, it is reversible. It does not bind permanently to the active site. It does bind to the active site, but reversibly. That's the essential feature. And it can be reversed by increasing the substrate concentration. That is exactly why the answer is B. A is wrong because it's not permanent. It does not change the structure. The substrate and inhibitor are the same shape. Actually, not the same. Similar. But that's not mainly the feature. It's not mainly a feature of the inhibition. The feature is mainly that it's reversible by increasing the concentration of the substrate. Batrachotoxin. It's a poison found in frogs in the Colombian jungle. The poison is to produce poison darts. The poison works by increasing the permeability of the cell surface membrane of nerve and muscle cells to sodium ions, which moves out of the cell. 4 students made statements about how the poison affects the cells. Water leaves the cells by osmosis, causing the cells to shrink. Okay, so this is a cell. It causes sodium ions to leave the cell. So Na plus leaves the cell. Water also follows. So yes, the cell does shrink. So I agree with 4.1. I don't agree with 4.2. This is wrong. It does not burst. So 1 is correct. 2 is wrong. When the sodium ions move out of the cells, the intercellular fluid has a more positive water potential than the extracellular fluid. That is true, which is why the water leaves. So 1 and 3. I'm going to go with 1 and 3. When the sodium ions move out, so the extracellular fluid has a more positive water potential. Wrong. 4 is wrong. Which process uses energy, ATP? Endocytosis, exocytosis. These are active processes, 1 and 2. Facilitated diffusion is passive. 15b. An indicator is colorless in acid and pink in alkali. Polyphenolphthalein. In an experiment, a petri dish of agar was prepared using an acidic solution of this indicator. A disc of agar 1 centimeter in diameter was removed from the center to create a well. A white card showing circular marker lines 1 centimeter was placed underneath the petri dish. So we had a petri dish. A disc of agar 1 centimeter of diameter was removed from the center to create a well. Then we placed a white card. Then 1 cm of alkali was put into the well, and the stopwatch was started. So it's pink in color currently since it's alkaline. A circular disc of pink color appeared and spread through the agar. It reached the first marker line in a short time but took longer to reach the second marker line, and a very long time to reach the third marker line. What explains these observations? So initially, this solution was acidic, which is why it was white in color. But then over time, the pink color we placed in the middle, the alkaline solution, spread over time. Okay, so obviously it's not facilitated diffusion because there are no carrier proteins involved. It is simply diffusing. And since the pink color is spreading, the indicator was already there beforehand. Okay, the pink color is spreading. This means that the alkaline solution is spreading. This is the only possible explanation. Because over time, the pink spread. Okay, the indicator was already there everywhere in the whole petri dish. 17. The diagram shows a section of a glycoprotein molecule found embedded in a cell surface membrane. Each of the amino acids is represented by a small shaded circle. Which row shows a property of the amino acids found in the alpha helix and a property of amino acid Q? So basically, this region is hydrophobic. It's non-polar, essentially, this region in the middle. And this region outside is basically polar because it's pointing to the water. Okay, so this section in the middle is hydrophobic. The alpha helix is hydrophobic. So it needs to be non-polar. And the amino acid Q is actually polar since it's in the water itself, okay? By the way, I was thinking, guys, how was your last year as an A-level student? How were your teachers? Did you self-study? Like, especially for, teachers are very good at explaining topics. But I think there's always a deficiency in people explaining paper 1 questions because the mark scheme only has the solution, right? I was just wondering where you guys get the solutions. Because back in the day when I was an A-level student, it was pretty hard to get the solutions. Like I had to think a lot. I was stuck most of the time, but took some time to get the answers. Hopefully I can help you with that. Some cells are listed. Which cells can divide by mitosis? Bacterial cells divide by binary fission. So 1 is out of the question. It's between C and D. The cancer cells can do that, okay. Lymphocytes can also do that, cloning. RBC do not have nucleus, so 4 is wrong. The answer is C. Stem cells can do that. Which statements about mitosis are correct? At the end of telophase, 2 nuclei are formed. Centrioles attach chromosomes to the spindle during metaphase. Chromatids are pulled apart during anaphase. 3 is correct. Chromatids are pulled apart during anaphase, just after metaphase. At the end of telophase, 2 nuclei are formed. This is also true. After telophase, cytokinesis occurs and 2 nuclei are formed. See this? Closed it by mistake. Here you go. Nucleolus and nuclei reform. This might be my last video for some time. I'm gonna get quite busy. My second semester just started this week actually. I'm still gonna try to drop some content. The October-November papers come out in January. So before then, I still have to finish some of the May-June 2022 content. I have some paper 4s, paper 2s, and paper 5s left, and the math papers. I'll try to complete those as soon as possible. So if you've been following my channel for AS, please support the channel for A2. That would help me a lot. So 19, yeah. So at the end of the telophase, 2 nuclei form. This is correct. Centrioles attach chromosomes to the spindle during metaphase. This is wrong. This is wrong. 19C. Check this. Basically, according to this question, centrioles in the centrosome attach chromosomes to the spindle during metaphase. So the centrosomes move to the opposite poles where they form the poles of the spindle, and each centrosome reaches a pole, and the centrosomes help to organize production of spindle microtubules. The spindle actually attached to the centromere. Chromosomes line up across the equator of the spindle. They are attached by their centromeres to the spindle. Okay. So this should actually be centromeres, attached chromosomes to the spindle. Okay. Centromeres. That's the wrong part over here. So that's why it's 1 and 3. So this is the chromosome. This is the centromere. The centromere of the chromosome. The centromeres attach the chromosomes to the spindle over here. Okay. Which statement about telomeres is correct? They allow cells in culture from any age of donor to divide a fixed number of times. Telomeres are at the ends of chromatids basically. They're genes which are present on the 5' end of every chromosome, unpaired regions of DNA on the 3' end of every chromosome. They prevent introns and exons being lost from genes during cell divisions. Honestly, telomeres just prevent aging. That's their process. If you want a more clear-cut explanation, here you go. The copying enzyme can't really run to the end of a strand. It stops a little short of the end. So we actually lose genes over time. So what is the main function of the telomere? It is to ensure that the ends of the molecules are included in the replication, not left out when DNA is replicated. Telomeres are found at the ends of chromosomes. They have been compared with the plastic tips at the end of shoelaces. They are made up of DNA with short base sequences that are repeated many times, multiple repeat sequences. They work by making the DNA a bit longer. They have no use of information but allow the copying enzyme to complete copying all the meaningful DNA. As long as extra bases are added to the telomere during its cycle, which is mainly done by telomeres, so they top up the telomeres. So according to this, the best possible answer, the single best answer is D. They prevent introns and exons being lost from genes during cell division. They're actually present on each end, both the 3' and 5'. They allow cells in culture from any age of donor to divide a fixed number of times. Not really. That's not the best answer. 21. The time shows the stages of mitosis, which is the correct order of sequences. Okay. Prophase, metaphase, anaphase, telophase. Clearly, this looks like telo. This looks like anaphase. They're moving. So it is bound to be 5 followed by 2. The end will be 5 followed by 2, not 2 followed by, sorry, it's 2 followed, sorry, 5 followed by 2, right? Not 2 followed by 5. So 5 comes first, then 2. It's between B and C. Now between these, it's either 134 or 314. 134 or 314. So metaphase isn't really shown here, is it? So you guys just need to understand, during the nuclear cycle, basically, guys, the chromosomes condense. So clear, look, in 3, it is the least condensed state. So 3 has to come first. 21 should be C. So over time, it condenses. Prophase occurs, metaphase occurs, anaphase, then telophase, okay? So the least condensed phase is basically your prophase. I mean, this is not prophase really. This is actually interphase. So after interphase, we get prophase. It is the least condensed stage. 20. Which statement about the transcription translation of a gene is correct? The non-transcribed strand of DNA has a base sequence that is identical to the mRNA produced in transcription. Not really, not really, because DNA does not have uracil. mRNA has uracil. Get it? The template strand of DNA has a base sequence that is identical to the mRNA produced in same. It's no. It's complementary, okay, but not identical, and it does not have uracil. The non-transcribed strand of DNA has a base sequence that is complementary to the tRNA molecules required in translation. Okay, this is the word. So, identical is a no-go. It's between C and D. So let's see, guys. This is your DNA. You have a template strand and the non-transcribed strand. So let me clarify this from the course book, since you're gonna follow it. Like, where do you get this info? Check this. So you have the transcribed strand or the template strand and the non-transcribed strand. The template strand acts as a vehicle for mRNA production. Okay? So this is the template or transcribed strand. Using the transcribed strand, we are going to get the mRNA molecule. So the mRNA molecule is actually complementary to the transcript strand. Okay, mRNA is complementary to transcript strand. So after you get the mRNA, you're going to get the tRNA. So this tRNA, right, it's complementary to mRNA. Who else is complementary to mRNA? The template strand, right? That is why, take a look at this. This is very important. Initially, we had the template strand. It was transcribed to form mRNA. Then from the mRNA, we got the tRNA. Required in translation, essentially. So 22, A and B are wrong because identical aren't proper options. It's just that in C and D, you need to understand the correct answer from here. Okay, it's a bit tricky. So I'll help you. For example, the non-transcribed strand of DNA has a base sequence that is complementary to the tRNA molecules required in translation. This is absolutely correct. The answer is C. Because think about it, check this. Look at the, for example, if this is the template strand, what if it was A, T, C, G, G? This is the non-transcribed strand. It should be T, A, G, C, C. Now let's look at the mRNA. Okay, look at the mRNA. The mRNA will be U, A, G, C, C. UAGCC. Okay, so this is the mRNA basically. Now, when the mRNA stays here, suppose this is the mRNA now, UAGCC. Let's get the tRNA in. tRNA. This is basically AUCGG. So this tRNA molecule, who is it actually complementary to? Check it. This one is complementary to this one. Do you get it? Because AUCGG, and here the pink strand, the non-transcribed one, TAGCC. Get it? That's why the answer is C. Tricky. You need to know this. Also, the non-transcribed strand, sorry, the template strand is complementary to mRNA. Which statement about mRNA is correct? The primary transcript becomes modified by joining of introns. No, we get rid of introns. Splicing. The primary transcript is synthesized and then modified to mRNA in the nucleus. Right, splicing occurs in the nucleus. mRNA contains nucleotides containing the sugar. No, no. Ribose. The bases in mRNA are held together by covalent bonds. No. The bases are held together. Which bases are they talking about though? Nitrogen's bases, right? So basically mRNA, where are they talking about though? Like here the bases are held together by hydrogen bonds. The bases are attached to sugars by covalent bonds. But mRNA itself has phosphodiester bonds in between. So B is the best answer, that's true. mRNA contains nucleotides. C is wrong and A is wrong. The bases in mRNA are held together by covalent bonds. So they're talking about the bases. Bases are always held together by hydrogen bonds, okay? But mRNA is single-stranded, so that's why it's vague. It's very vague. It's not right. Yeah, it's very vague. Like they aren't telling us the location of the covalent bonds. Okay, so B is the best answer. 24. Diagram shows part of a DNA molecule, which is labeled correct. Remember, purines have double ring structures, adenine and guanine. So double ring with triple bond, that's guanine. Single ring with triple bond, that's cytosine. C is basically double ring purine, this is adenine. D is a pyrimidine, that's basically thymine, which is labeled correctly. C is adenine, so C is wrong. D is thymine, D is correct. So I'm gonna go with 24, D. Hopefully that makes sense, guys. Double ring structures are purines, adenine and guanine. Guanine has the triple bond, so A is G, guanine, and C is adenine, double ring structure, purine with double bond. 25. The sequence of bases in DNA coding for the first 8 amino acids in the beta polypeptide of adult hemoglobin is shown. However, in hemoglobin C is basically one form of hemoglobinopathies. There's basically a structural defect in the hemoglobin moiety. Okay, so genetic defect, which causes hemolytic anemia. It becomes this. What's the change really? Here, 1, 2, 3, 4, 5, 6 amino acids. CTC becomes TTC. Which changes occur to the amino acid sequence of normal hemoglobin to make it C? Basically, this C is converted to thymine. Okay, so CTC to TTC, CTC to TTC. The normal amino acid glutamic acid turns to lysine. 25 is A. In sickle cell anemia, glutamic acid turns to valine. Diagram shows a plant organ. Which letter correctly labels the xylem? So this is the stem, basically. So the answer is D. Remember, in the stem, for a dicot, the xylem is actually towards the center. In the previous variant, in my previous video, I was talking about this. I was talking about this specific picture. You need to memorize this, okay? Everything else will be fine then. In the leaf, remember, the xylem is pointing towards the inside. The cross-shaped region in the root is xylem, and in the leaf, it points upwards. Okay? Great. 27. The photomicrograph shows a vascular bundle found in plant organs. Clearly, Y is the xylem. X is the phloem. This is not a root, guys. This is not a root. No way, no way. One is wrong. Because in the root I just showed you, it's a star-like structure. The xylem is over here. And phloem is like in these sections over here. Okay. So this is not a root. Some of the cells in region X have very large numbers of mitochondria. This is true. Because it's involved in unloading. 2 is right. So region Y is made up of a number of different type of cells. Yeah, I guess so. Yeah, xylem is made up of different cell types. That's true. Like 2 is for sure true. I was a bit confused about 3. But since 2 is correct, it has to be option C, right? Region Y is made up of different types of cells, but it's mainly the xylem. So this is basically the vascular bundle of the stem. This is the phloem. This is xylem. Yeah. This section basically. Okay. If you want more details, it's in chapter 7. Honestly, the worst chapter. I used to hate this when I was a student. Where is it? Xylem inside, phloem here. We have lignified fibers over here. I gave you a more simplified figure basically. It's lignified at the end. Like this. Okay, so yeah, it makes sense. It's alright. Anyway, with changes to the water potential and the volume of solution phloem safety element occur when sucrose is moved from the phloem safety element to an actively dividing shoot tip. Okay, so sucrose is moved from the phloem sieve tube to the shoot tip. So since sucrose is moved, the water potential in the phloem sieve tube decreases. I mean, water potential in the sieve tube increases. Why? Mainly because you're getting a solute out from the cell. It's like losing ions. If you lose ions or if you lose solutes, basically water potential increases. And due to that, excess water will now flow out of the phloem sieve tube into surrounding areas. So the volume also decreases. Okay? So there will be an increase in the water potential due to loss of sucrose, and there will be a loss of water which results in a decrease in volume. 29. Which processes occur during the loading of sucrose into phloem sieve tubes? Protons are pumped out of the cytoplasm of the component cell into its cell wall. 100% correct. 29 is 1 is correct. There is a higher concentration of protons in the symplastic pathway outside the component cell. What? This is the component cell. Cell wall. H plus is pumped in here. Then H plus moves down the gradient along with the help of the cotransporter. So the symplastic pathway refers to the intracellular, the pathway within the cell through the plasmodesmata, through the vacuoles. So there is a higher concentration of protons in the symplastic pathway outside the component cell. So if you want to recall this concept, here you go. This is the simplest and apoplast. The simplest pathway is basically the movement within the cell. Apoplast is the movement within the cell wall. Okay. So clearly the protons are present in the cell wall. This is a cell wall. So this doesn't make sense. It should have been the apoplast pathway. So I'm going to go with B. Protons are unable to move back. Wrong. It is able to move back with the cotransporter. A cotransporter acts as a carrier for protons and sucrose. Perfect. Perfect. 1 and 4. 30. The photomicrograph shows a section through a structure found in mammals viewed using a light microscope. Interesting. So it isn't D because this is the endothelium. This is the endothelium, one layer thick. Okay. So the answer to this is basically C. Why is that? If you study this properly, this is tunica media and the lumen is very large. So this is basically a vein. So in tunica media of veins, you have smooth muscle and elastic fibers, but no collagen fibers. Okay. So no collagen. No collagen, it's smooth and elastic. To study this, you can go through chapter 8. This section over here. In tunica media of veins, you only have smooth muscle and elastic fiber, no collagen. Collagen is only the outer layer. But if it was an artery, if the lumen was much narrower, the middle layer would have all three, and the outer layer mainly collagen and some elastic fibers. Okay? Great. This is a vein. So W contains smooth muscle and elastic fibers. Last 10 MCQs then. Which statement correctly links muscular or elastic arteries to their function. Basically, muscular or elastic arteries. Elastic arteries are close to the heart. They need to withstand the pressure. And muscular arteries are close to organs that they need to supply because they can constrict or dilate. The aorta is an example of a muscular artery. Wrong. It is an elastic artery. It's close to the heart. 31A is wrong. Arteries further away from the heart are muscular arteries. That is true as they transfer blood at higher pressure. This is wrong. You need elasticity to transport at higher pressure. Elastic arteries expand when the heart contracts and then recoil as the heart relaxes to maintain pressure. Exactly. Muscular arteries facilitate smoother blood flow than elastic arteries as they will expand and recoil. This is wrong, guys. That's the purpose of a elastic artery. 32. What is the heart rate in beats per minute? Okay. So this is one cardiac cycle. One cycle takes 0.8 seconds. So think about it. The beats per minute. Basically, in 0.8 seconds, we get one heartbeat. So in 1 second, we're going to get 1 by 0.8 heartbeats. So in 60 seconds, we're going to get 60 by 0.8 heartbeats. Let's see. 60 by 0.8. That gives us an answer of 75. Unitary method, essentially. So I saw a comment in my video for paper 1-1. Someone was asking me for a detailed explanation of the cardiac cycle. So let's talk about it right now for a bit, okay? Come on. Let's go. So guys, let's talk about this. So what's the detailed explanation? Not much. I'm just going to talk about it for like 2 to 5 minutes. You don't need the phonocardiogram or the electrocardiogram. You mainly need this. So honestly, according to the question they'll give you, this is one cardiac cycle, approximately 0.8 seconds. You need to know about 4 events. This event is basically the, this is the atrioventricular valve closing. This event is the aortic valve opening. This event is the aortic valve closing again. And this event is the atrioventricular valve opening finally. Okay, you guys need to know this in details. So what's happening in this section actually, from this moment onwards, okay, to this section, the ventricle is full because it's in a closed space. Okay, in this region over here, the atria have just finished pumping blood into the ventricles. Now the ventricle is just about to contract. There's a very short time span where the ventricle is about to contract, and when that happens, pressure inside the ventricle increases, but it is just high enough to close the atrioventricular valves, the tricuspid and bicuspid valve, but not high enough to open the aortic valves. But suddenly, when the pressure crosses a threshold, what happens is the aortic valve opens and the ventricle contracts. Now the blood from the ventricle enters the aorta, which is why the aortic pressure increases suddenly. But over time, the blood decreases, and after a certain limit, when the blood pressure is lower in the aorta, the valves that were open will close to prevent backward flow. And now, again, you know about the pacemaker, right? The sinatral node. It will send impulses to the atria to begin contraction again, since the pressure in the ventricles will decrease significantly. And when the atria receives the signal and starts contracting, okay, by the way, right now the ventricle is empty. We don't have blood in the ventricle. Now the atria starts contracting again. But before that, there is a time span where the atria is actually, filling up essentially. Right after ventricular contraction, the atria is filling up, and then the atria contracts. Okay, the atria contracts, and then the cycle repeats again. Okay. So let's take a look at these diagrams now. So this is the region where both are closed. This is also the region where both are closed. Okay, and the ventricle is full here, but it's kind of empty here. Okay, so let's see. Look at the ventricle volume. It's full over here. Then it contracts to let everything go. But then, right after the atria starts contracting, blood enters into the atria and into the ventricle, and it becomes full again over here. And then it contracts again. So see if you understand these stages. These you don't need these. I've basically learned this in physiology in med school. You don't need these details, all right? You just need to know about the events and what's happening where, and you need to identify the lines. Okay, like which one is the aortic pressure, which one is the ventricular pressure, which one is the atrial pressure, what happens to the volume essentially. If they ask you when is the ventricle empty, honestly, this region. This is where the ventricle is empty. So it's full over here and it's empty over there. So I'm gonna correlate these phases with the picture over here. So look at this purple section over here, the purple section. This one. This corresponds to atrial contraction. The atria is contracting over here, which is why there's the increased atrial pressure. Now, this blue section over here, this is where, oh, by the way, during atrial contraction, the ventricle is filling up actually. Okay, so here, the ventricle is achieving maximum volume. That's basically this section, atrial systole. Atrial contraction, the ventricle is achieving maximum volume. Then we get isovolumetric contraction. In this stage, essentially, the ventricle is a closed chamber. It is full. The pressure builds up, so both the valves are closed, both this and this. And when the pressure exceeds a certain volume, the aortic valve opens. Okay, the aortic valve opens, and then stage 3 begins. Stage 3 is basically ventricular ejection. After ejection, when the pressure falls slowly, then to prevent backflow, the aortic valve closes again. This is called isovolumetric relaxation. That's stage 4. And here the ventricle is basically empty, right? It doesn't have anything. And stage 5 is atrial filling again, because now, guys, the aortic valve is closed, but the atrioventricular valve opens again, and blood from the atria and blood from the superior vena cava and the pulmonary veins enter into the atria. That's stage 5. So the pressure in the atrium slightly increase, and then a ventricular filling occurs, which is basically the last stage, stage 6. This is a natural process, honestly. Like when the blood enters from the superior vena cava, inferior vena cava, and from the pulmonary veins to the atria, naturally, due to the blood entering naturally, some of it will enter the ventricles, but just a bit. Okay, just a bit, and then that process will be extended. More blood will enter the ventricles in stage 6, and then the atria will contract. Okay, so most of the atrial and ventricular filling occur quite naturally. Do you guys understand? And that is the diastole. So basically the atrial systole occurs at the end. Okay, right here. Stage 1 is basically atrial systole, and the ventricular systole is basically stage 3 mainly, but it also takes that stage 2 part. So we get the lub sound. Okay, why do we get the lub sound? This is a very good diagram. This is amazing, honestly. You can study this diagram. We get the lub sound due to the closure of the atrioventricular valve, and we get the dub sound. Why? Due to the opening of the atrioventricular valve. Okay, there are more heart sounds, but you don't need to know those. So that's all, guys. So this is the time span for the atrial systole, and this is the time span for the ventricular systole over here. Okay, it happens right after the atrial systole until, look, until the basically, wait, let me show you. So the atrial systole corresponds to this region. It corresponds to atrial contraction, and the ventricular systole, it will occur just afterwards and until the aortic valve closes. So we can say it like this, like ventricular systole will occur right after the atrioventricular valve closes and until the aortic valve closes again after opening. See, it opened and then closed again. That's the region of aortic, the ventricular systole. And aortic systole just occurs till the atrioventricular valve closes, and it's a region just before, you're going to see that the pressure of the atria rises just before. Okay, so according to this diagram, we can fill that information up honestly. Like this is the region of ventricular systole, and this is the region for atrial systole, this small region. Okay, get it? This is the lub, this is the dub over here. Dub. Okay, 33. I hope that's clear. The one who wanted explanation. Sorry, I can't do another video. Hopefully that clarifies. I'll link this in the um, description. Okay. Which events occur during ventricular systole? Great. Basically, during ventricular systole, the semilunar, the aortic valves, clearly open. The semilunar valves open. The atrioventricular valves it has to be, okay, 3 is correct for sure. So any option without 3 is wrong. The muscles in the ventricle valves relaxes. That doesn't make sense, right? It's a contraction. So 2 is wrong. It can't be 2. So the option is C. So why is 1 right? The atrioventricular valves close. Right. Check this. During ventricular systole, right after systole begins over here, honestly, the semilunar valves will open, or the aortic valves will open, and the atrioventricular valve closes. That is 100% correct. Due to the increased pressure, right? Due to the increased pressure, to prevent backflow, the atrioventricular valves close to prevent backflow to the atria. Okay, so it's 1 and 3 basically. 34. Which reactions take place in a capillary in the lungs? Okay, in the lungs, carbonic acid is formed from carbon dioxide and water. Honestly, this is what happens in the lungs. This reforms to form H2CO3, and then this forms H2 and CO2, which we release to the alveoli. Then hemoglobinic acid breaks down to form H plus and HB. What else? Carbaminohemoglobin breaks down to form hemoglobin and CO2. What else? What else? What else? And hemoglobin plus oxygen combines to form oxyhemoglobin. Carbonic acid is formed from carbon dioxide and water. Wrong. That's the opposite reaction actually. 1 is wrong. Carbaminohemoglobin is formed from carbon dioxide and hemoglobin. That is also wrong. Carbaminohemoglobin actually breaks down. Hemoglobinic acid is formed from hemoglobin and hydrogen. No, that happens in active tissues, not the lungs. Hemoglobinic acid actually breaks down to form hemoglobin and hydrogen ions. Carbon dioxide and water are formed from hydrogen carbon ions and hydrogen ions. True. 1 is fine. Only D. Last 6. Which features are important for the process of diffusion of oxygen out of an alveolus? Okay. Epithelium is permeable to respiratory gases. Must. 2 is very important. We need 2. Moist squamous epithelium present. Substance to reduce surface tension. Honestly, 4 is not a part of the answer. Surface tension prevents collapse. Okay, it should be A because it is permeable to respiratory gases. It is 1 cell thick. The pressure forces red blood cells through the capillaries due to the high pressure. And yeah, the moist squamous epithelium helps to do so. This is very important. You will see this in the course book in chapter 9. Where is this actually? This part? I don't think this was something you knew beforehand. Basically, you can read this section over here. Like, how is the diffusion gradient maintained? Mainly by blood continuously coming in and air being ventilated continuously. That's the logic, honestly. I guess this is too. It might be too complicated for you guys, but remember that it can't be 4. So we get rid of these, and we need 2. It needs to be permeable. Okay. So blood pressure forces RBC through the capillaries. This is the alveolus. We want O2 to come in. This is the capillary. So it's permeable, all right. There's moist squamous epithelium. Squamous is very so permeable. Squamous epithelium is perfect for diffusion. Squamous means flattened epithelium. You will see that in alveoli and endothelium. And blood pressure does force the red blood cells through capillaries. Okay, so essentially here, squamous is the keyword. Okay. Next, last 5. A student viewed 3 slides at low magnification and high. Each slide was a section through a different airway. Okay. So to do this, you can just match this table over here. We have goblet cells and cartilage only in bronchi and trachea. Okay, nowhere else. You have smooth muscle and cilia in the bronchial, terminal bronchial, but afterwards you don't. So let's match it with this. Many goblet cells. 3 is likely to be trachea or bronchi. It can't be bronchial. 3 can't be bronchial. Understood? It has to be trachea or bronchus. Next, very few goblet cells. So goblet cells are only present on the trachea and bronchus. Okay. But here's the thing. Very few goblet cells, like in the initial part of the bronchial, which is just after the bronchus, few goblet cells may be present. Very few. All right. Now, cartilage. Okay. This is the determining factor. Cartilage is present in bronchus or trachea only. So let's see. Slide 1, it needs to be bronchus or trachea. But here's the thing, irregular arrangement. In trachea, we have a regular arrangement of C-shaped cartilage. So since it's irregular, it has to be bronchus. That's why the answer is A. See if you guys get it. If it was regular, it would have been the answer C. 37. Which terms can be used to describe the role of mosquitoes in transmission of malaria? So basically, it's a vector. The female Anopheles is a vector of malaria. That's it. Malaria parasite is the pathogen, right? It's a protoctist. So the mosquito is only a vector, merely a vector. Rheumatoid arthritis. This is an autoimmune disease. It is common in women. What is an autoimmune disease? Basically, it's a disease where the cell's own immune system fails to recognize the self antigens and creates antibodies against them. It is basically a type 3 hypersensitive reaction. Table shows 5 different monoclonal antibodies, how they work. Inflammation and swelling of joints are, we use the drug CTLA4. So let's see, inflammation and swelling of joints are symptoms of rheumatoid arthritis. The cytokine TNF alpha activates cells in the immune system leading to death of cells in the joint. It affects the joint, mainly joint pain, morning stiffness. Which types of MABs could be used to treat rheumatoid arthritis? Let's see. Binding to proteins on the cell surface and triggering the immune system. This would actually make it worse. Blocking molecules on the cell surface that inhibit T lymphocytes. This will also make it worse. The problem is the immune system, cells of the immune system, right? They're the problem. So if we block the cell surface, block the molecules on cell surfaces that inhibit T lymphocytes, that means T lymphocytes would be activated more, which would cause more inflammation. We want to reduce inflammation by reducing the activation of lymphocytes and cells of the immune system. Okay, so if you block molecules that inhibit T lymphocytes, that means more T lymphocytes will be produced, and this would make it worse. More inflammation would be caused. Blocking cell signaling receptors that trigger cell division. Okay, let's think about this. We're not sure, but 1 and 2 are completely wrong. 4. Binding to antigens on cell surfaces and releasing a drug. Blocking cell signaling receptors that trigger the immune response. 5 is clearly a good option because the problem is the cytokine TNF alpha, which activates the immune system leading to death of the cells in the joint. We attack our own cells in RA. So 5 is correct for sure. I need 5. Because if we block the cell signals that trigger immune response, an immune system will not be generated. So we need 5. And I told you that 1 and 2 won't be accepted. 1 and 2 won't be accepted. So the answer is D. So I was a bit confused about 3 and 4, but here's the logic. For 4, binding to antigens on cell surfaces and releasing a drug. This is how the monoclonal antibody works. Yeah, the drug can work, which actually prevents inflammation and swelling. So 4 is totally fine. What about 3? Why is 3 wrong? Blocking cell signaling receptors that trigger cell division. Basically, guys, cell division is mainly done by T lymphocytes and B lymphocytes, which actually result in antibody production. But you know, inflammation can also be caused by other cells, like natural killer cells, macrophages. So we don't need cell division for inflammation. There are other mechanisms of cell division. So just because you blocked a pathway that triggers cell division doesn't mean the immune response can be prevented. Okay, so 3 is a very vague way. So it's 4 and 5. They're the best ways. Okay. Next, a person's blood group is determined by the antigens present on the red blood cells. I talked about this in paper 1.1 and 1.2. Which blood can be given to a person with group A? So basically, for group A, it's important that we look at the antibody of the recipient. The antibody of the recipient. In the person with blood group A, the antibody is anti-B. So we cannot give blood that has antigen B on it. That is why you cannot give B group because this antigen would react with the anti-B. You cannot give AB group because this B antigen would react with AB. So you cannot give AB, you cannot give B. You can give A and O. O can be given to anyone because it has no antigens. Okay, 39 is B. AB can receive blood from anyone because it doesn't have any antibodies. O can receive blood from only O. A can receive blood from A and O. B can receive blood from B and O. Okay, that's the logic. But according to this graph, B cannot receive blood from A and AB because it has anti-A antibodies which may react with the antigens. And A can only receive from A and O because, or it can't receive A, B, and B because it has B antibodies which would react with the B antigens that it would receive from the donor. Last one. Some of the events during the primary immune response are listed. Phagocytosis of foreign macrophage and antigen presentation. Remember, this is a must. This must occur at the first stage. That is why B is the answer. Antigen presentation is a must. There are three antigen presenting cells: macrophages, dendritic cells, and B lymphocytes. Some T lymphocytes will become T killer cells. The T helper cells divides by mitosis to produce lymphocyte clones. A T helper cell with a complementary receptor binds to the antigen being presented. Okay, so one happens first, then after the antigen is presented, the T cell receptor, we call it the T cell receptor, binds to the antigen. So 4 comes second. So this T helper cell then produces many clones, okay? And then this proliferates other T killer cells, and then it also stimulates other B helper cells. That's how it works. It stimulates these by using interleukin 2 and interleukin 4 and 5 mainly. Okay, that's what the T helper cells do. Alright guys, you don't need that many details about immunology. I just did it last sem, so I know a lot. Okay, anyway, hope you guys like the content, and if you did, make sure to subscribe to the channel. I'll link the video for May-June 2022 paper 1-1 up here, and the video for May-June 2022 variant 1-2 down here, and the playlist for paper 1 up here. And if I've made any mistakes, please feel free to comment down below. Okay, see you guys.